Hello everyone, and welcome to Genetics and Genomics BioConference. A brief introduction about myself. My name is Vikram Devgan. I'm heading a biological research content business at QIAGEN and managing next-gen sequencing related products. I'm located at Frederick, Maryland, US. And today I'm going to talk about application of targeted next-gen sequencing in detecting actionable mutations using multiplex PCR-based approach. I will highly encourage you that if you have any questions during my presentation, please write it down in Q&A box, and I will get to that at the end of my presentation. Okay, first of all, some legal disclaimers. All the QIAGEN product which we will discuss today or which are mentioned in this presentation, all are for molecular biology applications only. They are not intended for diagnostic or prognostic or treatment. Having said that, let's start the presentation. Okay. If you really look into the DNA, it's a very basic building unit of the genetic information, which is implemented as or imprinted as nucleotide sequence or a DNA sequence such as A, C, T, G. And any change in this sequence, one can also call it as genetic variants or genomic variants, may lead to change in phenotype or physiology of living beings. There are multiple different kind of mutations or genetic variations, ranging from point mutation to insertions and deletions, copy number variation, to large structural change such as translocation, fusion genes, so on and so forth. It's very important to detect these mutations because the change in these mutations may lead to some genomic diseases such as cancer. There are quite a few interesting mutations or genetic variants are already listed. A few of them I have listed over here, such as point mutation BRAF V600E. It's present in almost 60% of melanoma cases, and if we take the example of EGFR deletion and mutation in kinases domain, it's available, we can observe that in 20% of lung adenocarcinoma cells. And then there is HER2 copy number change or overexpression of HER2. Then there is BCR-ABL fusion gene, which is present in almost every case of CML. So these mutations not only act as diagnostic or prognostic biomarker, but they also play a very important role in deciding the treatment for a patient. Let's take an example of PLX4032. This drug works with melanoma patients only when the BRAF V600E mutation is present. There are a few more examples here for targeted therapeutic agents which are correlated or associated with different mutations. And this list basically goes on and connecting different mutation as listed here in genes to specific disease and then to a specific targeted therapeutic agent. And there is basically, this list is basically increasing very rapidly, and thanks basically goes to next-gen sequencing, as it can generate a lot of data. This growing list of so-called actionable mutation, as these mutations which are listed here with the different genes, if you detect these mutations, they are always associated with follow-up actions, and so we can call these mutations as actionable mutations. And Since the list of these actionable mutation and the associated targeted therapeutic agents are growing rapidly, it basically demands a change or it basically demands a shift in a paradigm of cancer treatment. So if you really look into the way the cancer treatment is getting decided, there is a need to do not only a routine pathology analysis, but also the molecular pathology analysis, basically to define so-called actionable mutation profile. And then one has to really look into the both the information from our routine pathology analysis as well as actionable mutation profile, and then one can decide about targeted therapy for that particular case. And now the question is how we can define this actionable mutation profile, or what is the best method to detect these actionable mutations. If you really look into the different technologies which are available to detect mutations, there is PCR, and then there is liquid bead arrays. We have sequenome, and then came next-gen sequencing, which basically has revolutionized the way we are detecting the mutations. Now the next-gen sequencing, what it can do is it can detect multiple mutations in multiple genes and with multiple samples simultaneously. In fact, one single next-gen sequencing run can generate tons of sequencing data. In fact, you can do a whole genome sequencing, or whole genome sequence can be determined with a single NGS run. But the question is, do we really need the whole genome sequence, or is there a need to have such a huge information or a data requirement? To basically answer that question, let's look at into the different variants which you can detect out of the whole genome sequences. Most of them are basically, there are millions of common variants, and most of these variants has no effect whatsoever on the proteins. And if you really look for the mutations which are associated with, let's say, disease or signaling pathway or the biological processes, they are in reality is somewhere around hundreds. And when we are talking about actionable mutation, they are in fact even lower. They are even less than the few hundreds. In fact, the Cancer Genome Atlas studies has shown that there are less than 100 genes which are hyper-mutated in common cancers such as lung, breast, colorectal, and prostate cancer. And if we really look into the mutations and the genes which are associated with targeted therapy, they are usually less than 20 per cancer type. And so this basically shows that what we need here is we need to focus on our sequencing effort to a very focused set of genes which are biologically and clinically relevant to a particular disease, instead of doing a whole genome sequencing and gathering so-called non-significant data. So we have previously we have so-called concluded that okay, next-gen sequencing is the method of choice to detect multiple mutations simultaneously. And when one has to do a next-gen sequencing, there are basically three different choices available. Either you can do a whole genome sequencing, or whole exome sequencing, or the targeted DNA sequencing. Now, if we have to compare and find it out of these three options, which is most suitable for clinical utility, basically targeted DNA sequencing comes out to be a winner. Basically, what it does, it provides you a clinically relevant data, as you can select the genes which you would like to sequence, and you can select only a clinically and biologically relevant genes for sequencing. At the end, you will get only a clinically relevant data. Then this is the most cost-effective method when we are talking in a per sample basis, and the targeted DNA sequencing can enable you to do a deep, deep sequencing or ultra deep sequencing, which in turn enables you to detect very low frequency mutations. And when we are talking about somatic mutations, it become really valuable, and it required that the method should be able to detect low frequency mutation. And least at the last, but not least, it requires very low DNA input amount, and which is kind of really helpful when you are dealing with the precious clinical samples. So just to sum up these things, basically, the targeted DNA sequencing is really a method of choice when you are looking toward the application of next-gen sequencing in a clinical, site. And that's the reason why the QIAGEN has developed the GeneRead, our GeneRead DNA targeted panel, and it is basically a multiplex PCR-based approach, or a multiplex PCR-based method to enrich your gene of interest prior to next-gen sequencing. What it does, it's in very simple terms, it provides you the set of primers for any gene or any region or any set of genes in a human genome. And in fact, what we have done is we have pre-designed primer all over the human genome, and then you can pick any region or any gene into the from the human genome. The primers are basically divided into the 4 tubes. Specifically, the overlapping primers has been segregated into the 4 tubes. This is to reduce any non-specific effect on any non-specific amplification during the target enrichment reaction. QIAGEN has basically the largest collection of wet bench verified gene panels. We have panels, as of today, we have 14 gene panels available, and we have 14 panels ranging from our clinically relevant panels to a tumor actionable panel for solid tumors, and we have myeloid neoplasma for hematologic malignancies, and then we have 7 tissue-specific panels or a focus panel ranging from breast, colorectal, liver, lung, ovarian, prostate, and gastric cancer. We also have a cardiomyopathy panel. And in addition to this so-called small and focused gene panel, we have a large comprehensive panels such as cancer predisposition, comprehensive cancer panel, and carrier testing panel. All these panels has been developed, and a lot of work has been put to ensure that we include only the genes which are biologically and clinically relevant to any particular or specific disease. So what we have done here is we have looked into a multiple references database ranging from the Cancer Genome Atlas to National Comprehensive Cancer Network, Cosmic Database, Cancer Genome Census, OMIC, and ClinVar, including our proprietary Ingenuity knowledge base, and manually selected the genes which are biologically and clinically associated with that particular disease in that. Just to give you an example here, I have listed the genes and the mutations which we are covering for tumor actionable mutation panel. This is basically a hotspot panel which cover the mutations listed here, and these all mutations are basically originating from 8 genes listed here, and we have selected these 8 genes and mutations on the basis of NCCN guidelines, and all these mutations are associated with clinical, actionable clinical relevance, and that's the reason why they are there in this panel. Further, to expand the application of this panel, we have developed clinically relevant tumor panels. These panels not only include all the hotspot in actionable mutation panel, but also include mutations or genes which are involved in clinical trials, as well as identified through retrospective studies. Beside that, we have also included the complete genomic coding region of the important tumor suppressor genes such as PAK, 3CA, P10, STK11, and TP53. So basically, this panel, what it does, it will provide the opportunity to analyze all the clinically relevant oncogenes as well as tumor suppressor genes with a single experiment. And as I said, that all the 14 panels which QIAGEN has developed, all of them are wet bench validated, and they show the outstanding experimental performance as shown here in this table. The first column is showing the name of different panels. The second column is showing the number of genes in that particular panel, and the third column is showing the percentage of coding region which is covered in a panel. Then the second last column is showing the number of reads which are can be mapped to a targeted region, and the last column is showing the percentage of bases which are read at least 20% of mean coverage depth. And as you can see from this experimental results, all of our panels show of more than 95% of coverage and specificity. And uniformity is also more than 90% for all of our developed panels. And how we can able to get such a high performance from our panels, it's because of our more than 10 years of experience in optimizing chemistry and designing primers. Interestingly, if you really look into that, the average amplicon size of our primer is around 145 base pair, which make it usable, these panels compatible with any samples, even including FFPE. So if you really look into the FFPE, you'll know that FFPE samples generally have or usually have a degraded DNA, and the DNA fragment size are in the range of 150 base pair. And with our average amplicon size around 145 base pair, this the GeneRead DNAseq targeted panels is ideal for analyzing FFPE samples for next-gen sequencing. And besides, all the assays which we have designed, they have designed in a manner which is suitable for the application of panel. For an example, in an actionable mutation panel, we have a very deep tiling of amplicon across the actionable mutation, as shown here for KRAS G12, whereas the clinically relevant panel, which is more of mid-size panel, as it has 24 different genes. There we have dense overlap of amplicons. However, when we are talking about comprehensive cancer panel, which are very large in size and have genes in a range of 160 or 165 genes, there we have less overlapping amplicons. The reason of having a different strategies for different panel is to keep a balance between the coverage as well as the next-gen sequencing real estate, which become really handy because of the cost associated with next-gen sequencing. And the GeneRead DNAseq gene panels, they are sequencing platform independent. What it means that they are compatible with all the sequencing, all the next-gen sequencing platforms. Either it's the sequencer is from Illumina, such as HiSeq, MySeq, or NextSeq, or the sequencer is from ironTorrent, like ironPGM or ironProton. So even if you have multiple different sequencer in your lab, you can use a single panel, and that panel can work with all the different sequencer you have in your lab. And if you look into the protocol of this targeted panels, the protocol is very simple, and it consists of 3 simple steps. What one has to do that they have to add genomic DNA, usually 10 nanogram per reaction, and since there are 4 reactions per kit, so you need 40 nanograms per sample, and you add the genomic DNA and the master mix into a targeted panel PCR primer mix, which we will provide you, and then run the PCR. After the PCR, you pool these reactions into a one and do the purification using ampere beads. And then after the purification, this is good for downstream next-gen sequencing library preparation. Here, one thing to note that although you are starting with 4 different tubes for the target enrichment, but you are pooling them after the PCR enrichment and going on only with a single tube for library preparation. So you have to do only 1 library preparation per sample, and this become really useful when you are processing multiple samples at a time. Then it it will simplify your workflow as well as reduce the cost of your workflow. And if you take a step back and look into the overall workflow for targeted gene panels, it takes 4 days from sample to insight. Basically, in day 1. Basically, if you are dealing with around 12 samples, it takes 4 days to go from all the way from sample to your data analysis and interpretation. The day 1 involve isolation of genomic DNA. Here I have used FFPE DNA isolation process. Next step is DNA quantification and qualification of that DNA. This step is one of the very important steps in the workflow, as any variation in the DNA input or a quality of DNA will have very profound effect in our overall sequencing results. So therefore, to make sure that you will have a very consistent DNA quantity and quality going into the target enrichment, we have developed the GeneRead Quantumized kit. So what this kit is, it's a PCR-based kit which do three things just with a single PCR run. You can do DNA quantification and it also provides a QC score to show that whether your sample is good for next-gen sequencing. And based upon this QC score, it will also provide guidelines to optimize your target enrichment. And all these three specs you can just get with a single PCR reaction. And after this, the next step is the target enrichment using GeneRead DNAseq panels and the master mix. Day 2 involve the library preparation, and we have reagents to do the library preparation. And then in day 3, one has to do the quantification of prepared library and then prepare for sequencing. In this case, we have used the MySeq, and which take around 24 hours to complete one NGS run. And that's why on day 4 you are good to do data analysis and data interpretation. But we understand that although we have 14 different catalog products and all of those products are Workbench verified, we do understand that one size does not fit all requirement, and that's why we have introduced a custom panel for GeneRead DNAseq sequencing. And what it does, it provides you a flexibility to select any gene or any genomic region or any set of genes, and then you can also get a flexibility to adjust the amplicon length as well as adjust the tiling density of your amplicons. And at the end, you will have fully customized gene panel, and if you look at the turnaround time, it's still very fast. You can get the fully customized gene panel to you with 3 weeks after placing order. And in the next slide, I'm showing basically the application of this custom panels. As we know that there are around 375,000 women in US those carry the mutation in BRCA1 and BRCA2, and these women are at very high risk to breast and ovarian cancer. And here the knowledge of this risk basically allows you to do the prevention, and that's what in this case we need to do a population screening for complete coding region of BRCA1 and BRCA2. But when we are talking about the sequencing of a complete coding region of BRCA1 and BRCA2, there is one specific challenge with that because these genes contain unusually high density of repetitive element. And so, which basically hindered the 100% coverage of the coding region of these two genes. However, using our customization ability, we are able to design the amplicons which can cover 100% of BRCA1 and BRCA2 gene coding region. So this is the experimental performance or experimental result from customized BRCA1 and BRCA2 panel. As you can see that the data shows that we get still with the customization panel, we get very high specificity and high uniformity, and more importantly, 100% of coding region is callable according to GATK callable loci Walker. So that is showing the power of customization ability, where you can change the tiling region or change the amplicon length and improve the coverage of your region of interest. The QIAGEN also basically believes not only providing us a solution but providing a complete solution, and that is why all this GeneRead DNAseq gene panel comes with a simple and easy to use online data analysis tool at no additional cost. What we have is this is a web-based data analysis portal, and what you have to do is once you are done with next-gen sequencing, just take that FASTQ file and upload that to our web portal, and then define a few parameters such as which panel you have used and whether you want to do a single or matched tumor normal analysis, or you are interested in somatic or germline mutation analysis. Once you have basically defined these parameters, the rest of the analysis the web portal does on its own, and it spits out the final analyzed report in the basic VCF file, as well as final sequencing metrics like what is the specificity, uniformity, or coverage of that particular next-gen sequencing run on a sample-by-sample basis. It also provides you all the detected variants in your sample, along with the copy number alteration report. In addition to these, this online web portal also has a direct link for ingenuity variant analysis, which can help to interpret biological or clinical significance of the data. So what is this ingenuity variant analysis? And if you really look into that, the ingenuity variant analysis is the mean to do the biological interpretation of a detected variant. In fact, it enables the finding causative variant quickly with up-to-date scientific knowledge. What it is basically a web-based software which provides a smart interface to shortlist any biological relevant variants or gene. So let's take an example. If let's say you're starting with total variants from which you have detected from your next-gen sequencing, and then you can use ingenuity variant analysis and first filter out all the common variants, and then you can use the filters to enrich predicted deleterious variant with statistical significance and in a biological context. And what you will have at the end, you will have the mutations which matter the most, or which are most biologically or clinically relevant to your research. So this is basically all about the data analysis and data interpretation part of gene panels. Now, if we'll take a step back and look into the whole portfolio of our GeneRead DNAseq targeted panels, we have panels for actionable mutation, clinically relevant, as well as disease-specific panels such as for breast cancer, lung, colon, and prostate, so on and so forth, and comprehensive as well as custom panel. And these all panels basically fit a wide range of applications. You can go from all the way from discovery to detection, or in other way from all the way translational to clinical research using this wide range of targeted panels. But when we are talking about the clinical research, we have to basically look into that the panels which we would like to use for clinical research. We have to look into the accuracy, sensitivity, or precision and reproducibility of those panels. And to look into these for our gene panel, we have collaborated with 3 different labs. Professor Susan Akaki from UK, Professor Putner from Germany, Professor Sangalves from USA. And what we have done, we have collaborated with these 3 labs and designed the experiments to look into the accuracy, sensitivity, and precision and reproducibility of our gene panels. The data which I'm going to show in next few slides, all that data is generated through our collaborators and analyzed using a QIAGEN NGS data analysis web portal. So for let's start with the accuracy. So for defining the accuracy, what we have done, we have selected the 10 different specimen or 10 different samples of which are known to have BRF V600E mutations, and we have analyzed these samples using our tumor actionable panel. So these specimen has been selected from melanoma, colorectal, and lung cancers, and they are previously been analyzed by alternative technology, either HRM or NGS or digital PCR, to make sure that we have the BRAF V600E mutation. And then we tested these samples using our gene panels, our tumor actionable mutation panel. And it shows that the tumor actionable panel can accurately call all BRAF mutation in all the samples. And if you really look into the mutation frequency, the mutation frequency goes from all the way from 9% to 74%. So it has the ability to detect the BRAF mutations in a wide different mutation frequency, and it has quite a good argument with what has been reported before. Now, to move on into looking into the sensitivity, specificity, and reproducibility. For this, what we have done is we have used a control sample which basically contain a range of endogenous gene variants which are basically present at different allelic frequency in a genomic DNA. We picked the range all the way from 1% to 24%, and then analyze that sample basically in duplicate in 3 different labs using a different lots of clinically relevant tumor panel. And what we got, the data is here. So we got very reproducible, precise data from all the duplicate experiment which has been done in 3 different labs, and we can see that our panel, along with QIAGEN NGS data analysis portal, can reproducibly call mutation frequency equal to or more than 10%. And if you really look into that, the data is very reproducible and kind of consistent from even lab to lab. Though we were not able to make a call of mutation which are in very low frequencies, such as 1, 2, or 3%, and however they are not called, but they were present in a sample which we can find it out by looking into the raw reads. This is because we have tuned our data analysis software in a manner that you won't find any false positive. However, if you use some alternative data analysis tools such as CLC Cancer Workbench, you can able to detect these low frequency mutations also. And we have seen that if we use the samples which contain these mutations in frequency more than, let's say, 8%, we can call those variants using our gene panel. This shows that our targeted panels have very good accuracy as well as reproducibility when we are talking about detecting mutations at more than or equal to 8% or 10%. And now, basically, if we have to really summarize why one should choose the GeneRead DNAseq gene panels, answer is basically quite simple. It is an intersection of the different solution and addressing different next-gen sequencing challenges. We have the broadest collection of workbench validated panels. Right now we have 14 different panels which is developed in-house and workbench validated. And in addition to these workbench validated panels, we provide ability to completely customize a panel for your own research needs. And these panels need a very low DNA input requirement. Basically, you need only as low as 10 nanogram DNA input for one PCR reaction. And the panel work with any samples, including FFPE samples, because of our low average amplicon length. And as I've shown you earlier, not only our internal data, but also the data generated by our customers shows the outstanding experimental performance of all the panels. We get very high uniformity, specificity, as well as coverage using these panels. One of the so-called bottlenecks for next-gen sequencing is data analysis and biological interpretation. And our QIAGEN's targeted gene panel provide complementary data analysis and biological interpretation to solve that bottleneck for next-gen sequencing. And it worked for all the next-gen NGS sequencers. So let's say if your lab have a sequencer from Illumina as well as from iTorrent, still you can use one panel and which will work with both the different sequencers. It needs only one library per sample, which is different from other multiplex PCR-based target enrichment panel available in a market. And this one become really important when you are processing multiple samples. It will not only simplify your workflow, but also reduce our cost a lot. And last but not least, it has a short turnaround time when you're processing a sample to a library. In fact, you can go from isolated DNA to a prepared library just in one day. So now, looking into what is the clinical value of these targeted panels, I have to say that the targeted panels are, their application into the clinical research and applicability for the clinics is increasing fast, and we are seeing multiple publications on that scope, and there are going to be increase in the use of these targeted DNA sequencing panels for clinical utility. And just to give you so-called the note at the end, we know that molecular profiling will drive cancer management in future, and it will require so-called high throughput cancer genotyping. And clinical next-gen sequencing is coming. It's not yet there, but it is coming soon. And targeted sequencing will play a key role in the clinical next-gen sequencing because of its forward-looking technology and its ability to scale up and to provide the actionable information in that front. So that's basically all I have for you today. And what I will do now, I will look into your Q&A to see if there are any questions, and I'll try to answer as many as I can. So one of the question is QIAGEN has developed PCR arrays for Drosophila 2. So answer to that is basically no. Right now we have these targeted next-gen sequencing panels only for humans. Another question is, can the same primer sets can be used for so-called RNA sequencing or cDNA sequencing? So the answer is basically no. We have design these primers which are covering also into intron exon junctions to make sure we are bringing these into the we are covering intron exon junctions also. So if you will use only cDNA, maybe you will miss a few of the coverage which might be important for your sequencing. Okay, there's the another question is, can QIAGEN bioinformatics team can develop or design panels for non-human targets? So right now we are not exploring that, but it can be done. We can you can contact the QIAGEN technical support or QIAGEN sales staff, and they can help you in that. One question is QIAGEN GeneRead quantumized kit is RT-PCR based? No, actually this is just qPCR based. It is for quantification of DNA and looking into the quality of DNA. Okay, so question is, do you use adapter barcode for a different adapter barcode or a same adapter barcode for a different sequencing platform? So one has to use a different adapters for different sequencing platforms, and as we provide the panels which are independent of adapters, so you can choose these adapters during a library preparation steps, and that's a reason these panels can be used for any sequencing platform. Okay, the question is, are there any automated platforms that use QIAGEN GeneRead protocol? Yes, there are. We have automated platforms. If we are talking about QIAGEN library preparation, we have QIAcube, which can be used to automate QIAGEN library preparation on it. And I think that's pretty much it. Okay, so in the absence of any further questions, I would like to thank all of you for attending this seminar, and feel free to contact me if you have any further question even after the seminar. I will be happy to take those questions. Thank you.